Great. Thank you. Thanks for the invitation, and thanks, Kristalina, for the introduction. So I'm hoping, so I've been here now about a year and a half, and I have given a lot of talks since I've been here. So, I was very nervous about this particular talk because I'm trying to find something new or some material here, and I can already see that there's some people here in the audience who have heard me present. So, if if you're bored, hopefully there's still something new in here that might be entertaining for you. And one thing I will say as well is that although I have AI in my title, AI is underpinning a lot. This is not an AI talk. Machine learning is underpinning a lot of the analyses that we're doing here. I just couldn't fit it into the 45 minutes, what we've been developing in this context, along with the biology that we have been working on. But this is an important slide to put up because this is the team that's doing all the work, and usually I run out of time before I get to let them have their due credit. These are many of the students that I'll be working on the projects that I'll be talking about today. I'm going to try to give 2 or 3 snapshots of some of the work that we're doing. And we do a lot of work in various spaces. We are primarily, I'm a professor of genetics, molecular genetics here, and so my training is in quantitative and population genetics from a computational biology perspective. Before I came here, I was in Toronto for many years, and before that, Montreal. And when I was in Toronto, I was head of computational biology for about 10 years. And we do a lot of work on evolutionary biology, trying to understand gene bioenvironment interactions as it associates with disease and aging. We're now getting more and more into trying to understand the cellular processes associated with aging as well. So there's going to be a lot of that in today's talk as well. And we're getting into also how do we integrate some of the, and you'll see why, some of the imaging data that we have access to and trying to understand disease processes. So today, most of the work that I'm going to be talking about has been done by a fantastic MD PhD student that I have, Jasmine Kang, who's only in her second year, and she's already got a Nature paper that just came out on Wednesday last week. So it's pretty, I wish that happened to me. It didn't, but it's happened to her. And Nick Cheng, who's been instrumental in some of the work that we're doing on early detection of disease as well. So that's a bit of a background already. So as I said before, we do a lot in the population space as well as in the single cell space. Historically, we've been working on trying to understand, say, mutational processes as they accrue with age. One thing I'm going to be talking about at Fairmount today is a program called CanPath. CanPath, so I'm from Canada, and CAN stands for Canada. And it is Canada's population cohort. So if you've heard of UK Biobank, CanPath is our answer to UK Biobank. We've recruited over 400,000 participants, and we've been following them for about almost 17 years now, I think. And so it's instrumental in all the work that we're going to be hearing about today. And we've been, again, I'm going to talk about how we've been using it to study things like the environment and its impact on health. I'll be also talking about how mutations in the blood of CanPath participants accrue with age, and also asking who don't have those mutations, and does that impact health? And so that's really a part of our program around healthy blood aging. And of course, I'm going to be emphasizing how population cohorts and population studies are really critical for thinking about the prevention side of health, right? So early detection of disease. The best cancer that you can get is the one you never get. And so that's where we're spending a lot of our time using these tools as well. Okay, so I thought I'd start with this slide because we will be talking quite a lot about aging today. And I thought it would be important to think about this question that comes up all the time. What is healthy aging? What is it when you're studying healthy aging? Are you somebody who just keeps aging like you're an 80-year-old and you have the blood, say, profile of a 40-year-old? Or are you somebody who's, say, a resilient ager, right? And we'll get into the distinction in a minute. And you may not even see that there is a distinction. It could be that a healthy ager is a resilient ager, but I think those processes need to be distinguished because we're going to talk about those biological processes in a moment. So this is, how you might want to think about aging. So and we've actually had a talk not that long ago by somebody from, I think it was, she had a background in Google, who actually made a very nice distinction between the variation in the population in terms of how we age. Who are those people who are aging? We all age, but who are those people who say do not develop the same chronic diseases or the same number of chronic diseases? We'll all develop chronic diseases as we age. Um, and we'll be focusing on that today. And of course, we're all going to age. So that green line that you see there isn't real, although there are people who do believe that we can solve that. I was at the Precision Medicine Conference in San Francisco in January, and it was an interesting set of discussions there that we think we can almost, particularly in men, never age. And this is kind of how I'm thinking about resilient aging, because we're all going to be impacted by, say, an infectious disease as we get older, as we age. And how you recover from, say, that, or say, getting, being at risk of type 2 diabetes or a stroke, is going to vary in the population. And that's how we might want to consider resilient aging as well. And that piece that I talked about that Jasmine had that was published last week really speaks to what is that impact as you age of, say, an infectious disease, of, say, inflammation associated with, say, chronic inflammation. Does it leave a lasting imprint in some people and not in other people, and why? Okay, now, I would say two lenses or two ways to study aging that are generally in the field there. And we kind of are trying to get at it from both, but most people either do one or the other. So if you're somebody like in my department at the NDPH, and you're working largely on, say, UK Biobank data, you're really trying to work at population scale data, and you're using epidemiological tools, Mendelian randomization, sort of in the drinking water up there. And don't worry about it; I hate that tool. But it's those are the kinds of tools that those folks like to use. Big numbers allow us to try to understand why some people are more resilient agers and why some other not. And then there's the other side, and the people who work on, say, those hallmarks of aging, those cellular processes, those people working on mouse models, whatever. They're almost a different kind of community. And those communities don't tend to really interact or talk that much. And I think the important thing for us who are studying aging is to really get those two sciences to work more closely together, and ask those kinds of questions. What do we need to, say, translate what we're learning at the cellular and molecular level into the population space, and vice versa, right? So just really quickly, the population studies obviously have a lot of strengths, relying on big sample sizes, for example, I've just mentioned. Obviously, they have direct human relevance because you're looking at the people. You're not looking at a mouse model, for example. And of course, they have some blind spots, right? And those blind spots might be that they are largely correlative, right? When we're doing those epidemiological analyses, we're looking largely at associations. You really do need the stuff that's happening on the left to look at causation, to identify causation. Now, they might argue over at epidemiologists that we do causation stuff. Okay, but really, it's, you really need to be doing these cellular experiments, these mouse experiments, organoids, and so on. Okay, now, let's just do a quick overview of what population cohorts can do for you. So this is and I like to call them population laboratories, right? So, and I didn't actually come up with that. Terry Manolio, who is the director of the the Genetics Institute at the NIH, came up with that term. But the population laboratory, as I mentioned before, you're directly looking at individuals in the population, and you can look at things like who are, say, getting diseases, who are getting cancers. And if you build a population cohort and study well enough, you can look at those population studies, say, retrospectively, and understand try to determine what those early determinants of health might be, right? If you capture, like, the UK Biobank, the UK Biobank is more powerful now than it was, say, 5 years ago or 10 years ago, because people are now developing those diseases, but it captured all this important information at baseline. Same thing with CanPath. So if you now have people who are, say, developing cancers, right, you can go back into the biobank, ask, well, what were they exposed to, say, 15 years ago, 10 years ago? Is there something in the blood in that biobank that you could have used, or you can use, to see what might have caused that particular disease to have occurred. And in the aging community, I mean, there's some hits and some misses, right? And there's some, and so I think studying aging using large population cohorts is really helpful. I mean, we've used longitudinal cohorts, as I mentioned a moment ago, to try to identify what those exposures might be or lack of exposures might be associated with healthy aging. I remembered a fellow named David Goldstein, who used to be at Duke, and I think he's now at Columbia, collected a large number of centenarians. And his real goal was, I'm going to sequence the genomes of all of these centenarians and see if there are mutations that might be protective among them. And unfortunately, he didn't find anything, in fact. And even though he collected a large number of centenarians, it was still probably underpowered for that kind of analysis as well. Longevity genetics usually means GWAS, genome-wide association studies. The big hits that keep coming are ApoE, and they keep coming. We heard a talk this morning about how ApoE and Alzheimer's keeps coming up, FOXO3. And we're not seeing actually a lot more apart from that. And then, of course, we're identifying functional and clinical markers. And you can't go to an aging conference without hearing about clocks. Proteomic clocks, epigenomic clocks, DNA methylation. You can actually go and buy a methylation clock now if you want for yourself to see how you're aging biologically relative to your age as well. And a lot of this stuff comes from a lot of a lot of those commercial tests come from the science from these big population studies that I've got here on the bottom here. And so, as I mentioned a moment ago, these population cohorts have been incredibly important for capturing at-scale genomics, proteomics, and we've learned lots of things. We learned that as you age, your memory T cells will go up, your naive T cells will go up. Naive T cells will go down, and megakaryocytes will go up as well. So I'm just going to give a couple more highlights here in what we've learned from population cohorts. And some things that have come up, and I'm going to get into a little bit more, is how exposures and the exposome has an impact on your health. And a study that came out using the Kaduri Biobank, UK Biobank, and a few others was really trying to see how much of the variance can best be explained based on, say, usual risk factors like age is a risk factor for aging, cell counts, blood sugar, lipids, and so on. And those are pretty good risk factors, actually. And we've learned about those for a long time. The Framingham Heart Study was actually a great study that's still going on that was built out of Massachusetts, Framingham, Massachusetts, and where we've learned about a lot of those risk factors. And adding polygenics polygenic risk scores or genetics hasn't really shifted or moved the needle all that much. We've only maybe explained perhaps about 2% of the variation with respect to aging. You add exposomics in there, and you can see a 20 to 22% improvement in terms of the variation explained, right? So exposomics can mean a lot of things to a lot of people, right? It can mean lifestyle, physical activity, diet, the drugs you take, like pharmaceuticals, so I mean, the air pollution and so on. So now, having said that, a very important paper, and I'd be remiss not to include this, just came out in Science January, that made the argument that we've been studying the heritability of aging and longevity all wrong, and this was their evidence for it. So most of our heritability estimates for things tends to come from twin studies, and particularly for longevity. And I just grabbed these two slides two pictures from the paper. And if you look at monozygotic twins, and I think this one was from the Swedish cohort, they just said, simply, if you remove extrinsic factors associated with death that might be associated with one or the other twin, you will seriously improve your estimate of heritability. And they showed that same kind of observation, and that's why the correlation in blue there is so much more pronounced. That red are the people who have one twin or the other twin has died from, say, a car accident, let's say. If you remove that information from your heritability estimate, you will seriously improve what you think might be the genetic underpinnings of longevity. And they did the same thing with the Danish twin cohort, and that's a dizygotic twin cohort. And so the estimates are a little bit less, but still significantly improved. So the genetics of aging, I think, is still up in the air in terms of what we know and don't know about it. Okay, so that's a little bit of a preamble about what we can learn from population cohorts. I'm going to explain a little bit about what we've been doing in the population cohort space. So I mentioned CanPath, it's Canada's population cohort. We've been running it for almost as long as I've been alive, it feels like. And it's as I said, it's increasing in its power because it's been a longitudinal study. People came into the cohort largely healthy, on average around 55 years of age. And we've been following their health over time. This is the breakdown. This is Canada for the Americans in the audience. I was living in Toronto. I started this activity when I was in Quebec, in fact. And Cartagena was the only cohort we were studying at the time. And Cartagena is a great cohort because it's a French-Canadian cohort. And apart from the fact that Montreal is a fantastic city and the food is great, it really makes the genetics a lot easier, because it's largely French and European, and founded about 400 years ago. And there's only been about 400 years of genetic drift in that particular part of the population. As you can see here, we've recruited about 400,000 people. We're still recruiting in Manitoba and Saskatchewan, and I'm going to be recruiting in the territory soon. Now, one of the things that we've also been able to do with CanPath, and this is a program that I was part of more directly when it started, but it's still going, and I'm not part of it that much anymore. It's called CANU, and it stands for the Canadian Urban Environment Consortium. So for all of those CanPath participants, I know where they live, and I know what they get exposed to or have been exposed to through this great database that they've built in Canada called CANU. And so we can do things like look at, air pollution exposures, right? So that's southwestern Ontario. And for example, at a 3 to 6 digit postal code resolution, I can map out how people are exposed to, say, what do we have here? NO2 concentrations, or PM 2.5. So various measures of air pollution. As an example, I can look at green space. We can look at how much access do you have to proper food, road networks, and so on. And we can look at that longitudinally, and we can look at that in combination with how people age as well. And, what we've been doing, so that was Toronto. We have the same kind of data from Montreal. And in Quebec, we've got some really good data now on genomics, transcriptomics, which is our way of looking at functional genomics. We're now looking at proteomics as well. And we can start asking questions about how those molecular markers, as well as your genome, interacts with the environment and then potentially contributes to health risk. Okay? So I'm going to talk a little bit about this, and because it's one of my favorite projects. So what we did initially to really try to understand how genetics versus environment might impact, say, molecular determinants associated with health, is we collected data in biologics and functional genomic data from 3 places: Montreal, Quebec, and the north of Quebec, which is a town called Saguenay. And actually, it's a very nice place, Saguenay, Lac-Saint-Jean. But for these purposes, I've put the aluminum plant that's there, which has an impact on the environment. When you're there at night, you can really see the change in the sky color. It's got a different kind of hue than if you were, say, 500 miles away from that. And one of the reasons we picked these 3 different places is because when we did some genotyping, what we were able to capture was, in fact, so here's just the measures of the environmental variability. But when we did some genotyping in Quebec, the picture on the left says genetic ancestry. So that's effectively a principal component plot, coloring the different postal codes. So I've colored the individuals based on a principal component of their genotypes, right? And you can see there on the bottom, your left side, is that people in Montreal have a slightly different genotype pattern than people in Quebec City versus those in Lac Saint Jean Saguenay in the north, which is a little bit surprising considering that all of these people came over from France 400 or 500 years ago. But what it tells us is that it was like a sub-founding population event. People largely came in from France to New France, landed in Quebec, and then went either north or to Montreal. All right, and you get these sub-founding populations occurring, and that's to some extent what we call population bottlenecks. You can see the different, when you measure, say, inbreeding, take inbreeding measures or consanguinity measures, which is a nicer word. The genetics term is genetic inbreeding, but you can really, that probably has a significant impact as well. The proportion of secondary cousins in the north of Quebec is actually higher, much higher than, say, in Montreal. Now, when we looked at gene expression, so, okay, that's the genotype data, right? And then that's the environment data. And as I mentioned to you before, the environment looks very different in those three different parts of Quebec, right? So blue is same principal components, but I took PM 2.5, which is a measure of air pollution, and you can see that's different as well. And then we looked at gene expression data, and we also saw differences there as well. So gene expression, for those who aren't completely familiar with functional genomics, is really a measure of how much genes are turned up or turned on or turned off, okay? So that seems to also vary. So the question then is, what is driving that variation in gene expression? Is it the genetics or the environment? So the way we tackled that is by using a neat little tool that we have, because we can not only see, okay, so let's start with this. This is a volcano plot. For those who aren't comfortable with a volcano plot, it's pretty straightforward. They look like volcanoes. They go up. And generally, the effect size is what's being plotted on the x-axis, and on the y-axis, you've got the p-value of each dot, which is a gene, right? So what I'm measuring here is how differentially expressed a particular gene is in two different environments, right? Depending on whom I'm comparing, right? So on this plot here, I'm looking at people from different environments with different sub-regional ancestries, right? And you get lots of differences in gene expression. Okay, not too surprising given what I just told you. Then in slide B, we've got people in different environments but similar regional ancestries. So the reason we have similar regional ancestries is because I know people's genotypes. I know what if your genotype makes you a Montrealer versus a Quebecer versus a Lac Saint Jean. So if you were living in Montreal and then moved to Lac Saint Jean, I can tell that based on your genotype. So that's how I can say, look at people who are in the same environment or in different environment, same genetic ancestry or different genetic ancestry. Does that make sense? And so when we have Montreal locals, different environments and same regional ancestries, you still get those volcano plots. You still get those reds, which means that they're significant. You get significant p-values for those particular genes. Okay? Now, if I do this analysis where I look at the same environment but different genetics, right? So people who move from Montreal to Quebec, so they have the same environment, and I compare people within Quebec City, regardless of your genotype, you don't get the explosions, right? You don't see differential gene expression. So I'm controlling for genetics here and being able to evaluate the exposure. And what you're seeing here is that it really is the environment, not the genetics, that's driving your functional genome, how genes are being turned on or off, okay? So speaking to that point that I made a moment ago about that Kaduri and UK Biobank study where they said 20% of the variation versus the genetics explains your risk of things, this also speaks a little bit to that as well. And what is it about your environment? Well, here we're just going to blow this up. The three different colors in that dendrogram are just where people are from, and you can see really what's being lit up are things associated with air pollution as well. And I don't have time to get into a lot more of those. More of this data in terms of the impact on lung cancers. I will say, though, that we had a paper with Charlie Swanton from CRUK where we used that information to show that those types of that type of exposure leads to a rise in inflammatory signals, which we think really promotes lung cancer in non-smokers, both in Quebec, Vancouver, and in other places across the world. Okay. So I'm going to switch gears, but I'm going to still focus on blood. And I'm going to move away from exposures of the air pollutions kind of perspective, and now get you thinking about early detection and early prevention. And I know that we've already had maybe a talk or two about early detection. We're also in this space, but we're in this space from the perspective of, and maybe this is a question we can talk about afterwards. If I were to tell you that I can detect a cancer 9 years before, say, a prostate exam or a mammography, and give you a risk profile, what would you do with that information? And generally, that's what this work is all about here. So we all know that if we treat cancers and treat diseases earlier, thinking about it through a healthy aging lens, you really want to be thinking about, again, that idea that it's the best cancer is the one you never get. So the earlier we can detect a cancer, the better the interventions that we have will work. And these are just some of the standard types of screening tools that we have in the population level. And I think you'd be living under a rock if you haven't been hearing about some of these blood-based tools that are out there, where people, Grail is sort of like the famous one that's out there, as largely famous because of the amount of investment they have, but taking a blood sample and making a claim that they can see a signature of cancer. Now, most of the tools that are out there do work, but they largely work in the context of you've already been diagnosed with the cancer, you've been treated, and we're going to track you in cases for remission. What we're working on here, and it is how do we use those big population cohorts like UK Biobank, like HandPath, take those individuals who've never developed a cancer and see if we can see a signature there. And the tools that we're developing could work in the context of cancer, but they don't have to only be cancers. And anytime there's any sort of damage in the body, you're going to see shedding of material, and it's going to end up in blood. So you just have to, say, modify some of what your signatures or markers that you're looking for, and you might start seeing things like, you wouldn't type 2 diabetes if that's really what you wanted to look for, but there are better tools for that. So you wouldn't maybe type 1 diabetes, autoimmune, brain-based diseases as well. So we've been working in that space, and I'm just going to move a little bit more quickly because we're already running out of time. Yes? All right, we're running out of time. Okay. And one of the tools that we've been developing is it's a DNA methylation signature. Now, the reason we like DNA methylation is because every part of your body leaves a specific DNA methylation imprint. So if we can see in plasma, for example, a signature of damage and cells are being shed into the blood, that methylation signature we can use to discriminate the methylation from blood versus, say, something that's coming from, say, your pancreas or something like that. And those are the tools that my student Nick has been developing. And what we've been doing is leveraging from this is a map again of Ontario, just from Ontario so far, bloods from people who had never developed a cancer. And because they've all consented to allowing us to link to their administrative health records or their electronic health records, we know who's gone on and developed a cancer. So we can retrospectively go back and pull from the cohort the bloods from people who have been diagnosed with cancers. And we can do that for various time points. We can do this from you were diagnosed, unfortunately, a month after you gave us a blood sample and signed up to be part of the Ontario Health Study, or maybe 9 years later as well. And you can see here our numbers for our first pass of the data are highest for breast and prostate because those are the most prevalent cancers in populations. Pancreatic cancer is something we'd like to really dive into because it is a disease that once you're diagnosed, it really you're usually diagnosed at stage 3, stage 4. And so if we can make a dent in terms of early detection there, that'd be great. We still have a problem with numbers, though. And so population cohorts may not be the solution for us here. But again, these are some of the numbers. And so so you can understand the picture on the bottom, I've just normalized everything on the left axis. When the participant came in, we just said it was time 0, and going back in time is when they were going forward in the figure, is when they were finally diagnosed with the cancer. These are the kinds of data that we have for these people. Because in Canada, we have a pretty good screening program for mammographies. And it works pretty well. Most people, when they're being detected, they're stage 0, stage 1, right? And so you can kind of get a sense that the data that we have, when they are detected, Most of those numbers are stage 1 and 2 for breast cancers, right? Still, numbers are there. Prostate may be a little higher. And you can, that point that I was making before about pancreatic cancers, many more stage 2, 3s, and 4s there in the data. And I'm just going to go through all the pictures of sequencing machines. And generally, again, here's our volcano plot for those who are now more comfortable with those types of analysis. But instead of gene expression, I'm now looking at differential methylation. So every dot here would be a feature in the genome that has been methylated. And I'm asking for those patients those people who are now patients, whether there's more methylation in their plasma than against the control. And the neat thing about having population cohorts here is I can really control for other factors as well. I can control for age, BMI, alcohol use, smoking use, and so on. So we have a lot of control participants in these types of studies as well. So our first pass of the data actually did pretty well. We have different colors of dots for breast, prostate, and pancreatic, depending on how far back in time the blood sample was collected. And you can see already that the PCs are showing us that there's some good separation between cases and controls. So that encouraged us to go back into the cohort a couple of years later. And the numbers are exponentiating of people, because that's how cancers work. The number in terms of prevalence, they will exponentiate over time. And this is just breast cancer over here. And I will say, by the way, everything that I'm going to, this is about to appear in Cell Genomics in about 2 or 3 weeks. So if the details aren't readily forthcoming for you here, then you can dive into that there. The difference here that I have, though, is I got my colored dots of yellow and of red for cancer diagnosis, black for sample collection. But I also have information about when did they have a mammogram. So I can now ask, how well does my blood test work compared to a standard screening approach, like a mammogram as well? Is there something the blood test caught that the mammogram didn't catch? It would be the question that we can ask. So when we look at this for just breast cancer, again, we're getting more samples. We're doing pretty well there. We use some pretty standard now machine learning tools to make sure that we're not overfitting our models. And generally, these are how well our tools are working for breast cancers. And in many cases, we do better for certain kinds of breast cancers than others. Triple negative breast cancer and early onset breast cancer, we're around an 80% accuracy. For those that are a little more later age of onset, and this really makes sense to me because we think the biology here is different of these different cancers, is different and it has an impact on our ability to detect these things. Survival plots for those who are interested in. This is how we're doing for prostate. Same kind of data that I just showed you a moment ago. So for primary prostate, we're actually knocking it out of the park. Our accuracies for detection are almost 90 to 95%. So I think that's first in class. We can also see that for metastatic breast cancer, we do very well compared to looking at, say, early detection or primary. Breast cancer is the tough one. All right, and even though it's got the biggest sample sizes, we're all struggling to try to improve what we can. And we have some new evidence that maybe think that we're looking in the wrong places, actually, for breast. But I think we can do better there as well. And the last thing that we can do with these tools, finally, is look at things like exposures. How is alcohol frequencies impacting our scores? How is smoking impacting our scores? So for here, I'm looking only at prostate cancer. So if I include alcohol frequency in my model, coupled with my methylation risk, I actually improve my probability, my probability for detecting a cancer, which is also implying then that alcohol is a significant risk factor. And in Canada, the Canadian Cancer Agency, has already said that publicly, and this was actually made a big noise. No more than 2 drinks a week. It's a carcinogen. There's already discussion about maybe putting labels on things like the way we put labels on smoking. So, okay, and lastly, we had a piece in Nature Cancer where we have a whole compendium of how these types of signatures look across a whole bunch of cancers as well. All right, now the last thing I'm going to talk about today, so I think I'm not actually too bad on time, so thank you. let's get away from some of the population stuff, and now let's start moving towards some of the more cellular stuff. And hopefully, you'll believe me by the end of it that I've integrated the cellular and the population to think a little bit more clearly about healthy aging in particular. All right, so I think I mentioned this briefly already, that when we're studying aging from an animal model or cellular model, we're looking at a different kind of lens, right? But those lenses have been really important for us to understand, say, what we call the hallmarks of aging. And the hallmarks, there's a hallmark for everything now. There's a hallmark for aging, there's a hallmark for cancer, there's a hallmark for cardiovascular disease. And those have largely been built from things like model organisms. We've learned from those programs that telomere degradation has an impact on aging, epigenetics has an impact on aging. And from those programs, we've also learned about things like interventions, like senolytics and so on. So this is what the standard picture from Lopez Oten in 2013, and it hasn't really changed. Every 2 or 3 years, they come up with a new hallmark of aging. But these are the omic features, if you like, or the cellular features associated with aging. So as I mentioned, genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, those are one kind of, in my group, we kind of think of them as a certain kind of class of aging traits that happen at the cellular level. Things that may be more associated with response to damage might be around, say, mitochondrial dysfunction or senescence. And then there's other things associated with just more the aging phenotype, like stem cell exhaustion. We tend to see that more in blood in particular, and altered intercellular communication. Altogether, these things speak to what we call the hallmarks of aging. Now, most studies that I just talked about at a population level, we don't really discriminate any sort of tissue specificity. Like when you hear UK Biobank give you a talk, they've only captured blood and plasma largely, and they're going to say lots of stuff. This is not. I'm not being negative about UK Biobank. That's not how this is meant to be. But largely, you can't make some of the claims that you might want to make about, say, how the brain ages based only on one tissue, which is what they have, which is blood. Right? However, others have tried, right? And from looking at, say, the proteomic clock, some people have tried to make an inference that an individual will age at very different rates depending on what tissue you're looking at. Right, so your immune system or your blood system might actually be aging at a much faster rate than, say, your lung, right? Or your liver might actually be quite young looking compared to your brain aging or your blood aging as well. So people are now really trying to build proteomic clocks based on blood. But it's not the same thing as doing a deep dive into the cellular biology of aging, of the tissue you're interested in. So we're going to stick with blood for now. And we think blood is a really important it's an important phenotype to look at. Immunoaging and inflammaging, you may have heard of those terms before, but those are critical features associated with aging. And of course, blood is important because it touches all of the organs, right? So as I mentioned before with the cell-free DNA work, it captures lots of different things. So we decided, because I run a biobank and I have access to other biobanks, we're just going to focus on blood aging. And let's think about this from the lens of just the blood and try to be agnostic about it. Like, how do we select our participants to look at blood aging? Right. So we found this thing called the Intermountain Risk Score, called IRS. And it was built by the people who are at the Intermountain. It's a health, it's like Geisinger in the U.S. They're largely out in Nevada and California and so on. So they provide health services to people in those states. They built this 5-year mortality risk score, and the variables that go into the Intermountain Risk Score are largely just cell traits, right? You can include age, you can include lipids or electrolytes or whatever, but it's a really cool tool because I can take them out and can still see what actually drives healthy aging. And for them, it was just these different, and when I say cell traits, I mean neutrophils, red blood cells, white blood cells, those types of counts. And so what we decided to do was make a derivative of the Intermountain Risk Score and called the Complete Blood Count Risk Score, so CRS maybe, or CBC, whatever. And what we did with that score is we went back into CanPath, and I selected participants based on different ages and different CRS scores or IRS scores, whatever you want to call them. So the idea here is to find people who are aged but low risk. So who are those people? Who are those 80 or 90-year-olds who have the blood profile of, say, a 40-year-old, right? And let's really kind of interrogate the heck out of those blood samples and compare them to high-risk individuals. So AH is a high-risk aged individual. AL is a low-risk individual. So you can see here the CRS is on the bottom, which is my low to high mortality risk on the x-axis, and I'm looking at the different age groups. And I wanted to do the same thing for young people, so young low risk and young high risk, and pull the blood out of the biobank from those individuals, making sure I'm not selecting people who, when they came into the cohort, had a chronic disease or a cancer, or a chronic disease, cancers, we think of the same thing, or other sort of unfortunate events like that. And of course, capturing also all kinds of other information about BMI and smoking and so on. And so one of the things, and I think this is actually quite striking, before we got into any sort of molecular genetics and biology, if you just look at the blood traits on this picture here, so I'm looking at hematocrit, white blood cells, platelets, MCV, MCH, and RDWCV. Just focus on RDWCV, which is red blood cell, coefficient of variation of red blood cell width. That's what that means. And these are all the standard things you get when you get a CBC, a blood test at a clinic. And you can see that RDWCV by itself is actually pretty variable and almost predictive of those 4 different risk classes. So I've got those 4 different colors of violin plots there. They're the 4 different AH, AL, CH, Y, whatever. You remember. I'm trying to get this done in time here. And you can see that RDWCV is actually more similar in the unhealthy groups versus the healthy groups, regardless of age. So the question there might be, why? And actually, if you go to a very large Israeli study that has access to the entire Israeli health record, they can recapitulate that as well. And there's a biological explanation that we'll get back to that in a minute. So what we then did with all those blood samples is we had live blood, so we did single-cell RNA sequencing of all of those participants. And to date, and we're about to get blown out of the water by UK Biobank, but this is the largest population cohort of single-cell RNA sequencing coupled with genomics, coupled with epigenomics and attack seq and so on. And then we wanted to ask simple questions like, at the cellular level, what is driving the variation between these high-risk, low-risk, young and old people? Okay. And one of the things we were able to do is build a transcriptional risk score and identify different genes that are differentially expressed. The transcriptional risk score is effectively just taking those volcano plots that I showed you earlier, combining that information, and identifying genes that we think are driving variation in health between individuals. And what this plot is not only trying to show is that observation, but it also varies tremendously for males and females. So I've got mean coefficient of female effect sizes on the x-axis, and on the y-axis, I've got the mean coefficient of effect sizes of males. And you can see if the things are on the x-axis or on the y-axis, that means that they are sex-specific. They are male-specific genes and female-specific genes in specific cell types as well. And if I put all of that information, the headline is largely what drives a healthy aging male, or a healthy ager in general, is a maintenance of similar gene expression to that of a younger person, right? So a healthy ager generally has maintained the gene expression profile of a young person who also has no disease. However, we were able to find specific factors. So most of the variation that explains healthy aging is driven by a maintenance that these people manage to maintain their gene expression profile. But in a few instances, we found unique events in those healthy agers that might actually have had an overall effect on their overall gene expression profile, that were distinct in those healthy agers. And in a way, this figure is not doing a great job of covering it. I'm going to jump really quickly here. If we go back to our hallmark of aging picture, which of these hallmarks of aging are the ones that are coming up the most? Well, it's actually almost all over the place. But chronic inflammation and mitochondrial dysfunction are the genes that are highlighted the most. So those healthy agers have genes, expression patterns similar to those of a young person in those pathways. So really what this is pointing to is that chronic inflammation, more so than mitochondrial dysfunction, I want to emphasize that actually, chronic inflammation is a significant hallmark of, and your management of that somehow is a significant hallmark in our cohort of healthy aging. The last thing I'm going to mention, as well, I did mention that maintenance of gene expression is similar in healthy agers, was this study that we had just published, which really is getting to this point. This is this piece that was just published a few weeks ago, really, again, speaking to this inflammatory process here. In fact, this was a project that we weren't actually thinking was going to point to this, in fact, but we work in somatic aging and mutation and so on. So we had captured blood samples from Parrish Vyas's lab, who's at Weatherall, and he does all these bone marrow biopsies, and he gave us all these single cell samples. We got a whole bunch of single cell material from people working on sickle cell anemia, people pre and post COVID. So it was really what we were thinking we were going to be seeing was an impact of infection on the accumulation of somatic mutations. And the reason we were interested in that is because in my lab, we study how stem cells, your blood, accumulate mutations as you age. And we know it to be a significant risk factor for health and for blood malignancies, cancers. And so this is a phenomenon called clonal hematopoiesis. And similarly, you'd be living under a rock if you've not heard of clonal hematopoiesis. It seems to be associated with everything, specifically blood cancers, but we now know more about it in the context of dementias, certainly with chronic CVD. Sid Jaiswal, who's now at Stanford, has a seminal article in 2014 in New England Journal that really profiled just how important it is across a range of chronic diseases. So what we were initially looking for was how does maybe your somatic mutation profile get impacted by insult through infection? And we work a lot in that space. Oh yeah, there's Sid Jaiswal's paper right there, in fact, actually, where he shows, I think this is the total number of mutations, and this total number of somatic mutations that you'll see in your blood accumulate as you age. This is our work looking at where if there are hotspots of that in your genome, and this is our work looking at how that might vary in different populations. So we've been working with the H3 Africa folks to see if there are different hotspots of somatic mutations. And you can see, say, for example, on chromosome 5, there's a specific occurrence of somatic mutations on chromosome 5 in Africa that we don't see in other populations, and there's other hotspots of somatic mutations. And we think that might have something to do with infection. That's what led us to thinking about, actually, this whole project. And in fact, what we didn't see was an impact of inflammatory stress on somatic mutations. But what we did see is when we looked at single-cell RNA sequencing was that pre- and post-COVID infection was a new sort of population of stem cells. And we call it the IM population. It's the HSC-IM population. So you have, these are people that we thought were a COVID signature, and we saw it with our aging signature as well. As you age, you also see this new population that you don't see in a young population. And when we dive deeper into that population of cells in your bone marrow, we saw that there was also an epigenomic alteration. Chromatin is being modified or remodified at the chromatin level. That's giving us this different cellular picture at the RNA level as well. And the last thing I'm going to say about that is that when we looked at that in population cohorts, where we also have single-cell RNA sequencing, that whole healthy aging population cohort that I just told you about before, where we were able to look at low and high risk, blah, 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 that memory, that signature, we see in the downstream daughter cells of those people there in that cohort as well. So that is my marriage, if you like, of single-cell mouse work, bone marrow work with single-cell data from population cohorts. Those signatures that we were able to find at the stem cell level, we were able to see also in populations at the population level. So actually, that's just the summary of that as well. And so what we think is that that insult due to infection or to aging, associated inflammation, not only do we see it in all of your progenitor, your derived blood cells, but it actually, in some people, leaves an epigenetic imprint. And that speaks to that chronic inflammation story that I was just trying to get across as well. So when we go back to the hallmarks of aging again, chronic inflammation, those genes that came up in that study are also associated with the chronic inflammation pathway, also associated with stem cell exhaustion. And they also come up there, like as I mentioned before, there's a hallmarks of cancer, and we try to see what comes up in the context of hallmarks of cancer. So inflammation, bad. Okay, and that's pretty much it. So I'm going to end this presentation with thanking a lot of the people that have been important in the omics, in the data collection, the data analyses. Of course, this picture is entirely missing out on all the people. There's about 100 people that work at CanPath who do all of the really hard work in recontacting participants. We have a big nutrition and diet study happening. We have another activity right now that's going out and getting more blood from those people. All those people who haven't developed a cancer, we're getting more blood from those people. They, of course, won't have developed other types of disease as well, otherwise they wouldn't be able to consent. So that's all happening now. We're very excited about where that work, that longitudinal blood data will take us as well. So just to summarize some of what we've done, I hope we've convinced you that population studies are important. They allow us to look for early detections and early signatures of cancers. They allow us to look at the cellular studies with regard to aging, and they also allow us to look at the epidemiology as well. That's it. Sorry. Thank you.